Good morning, everybody. We are now continuing in our chapter 2 for biology 2. So in chapter 1, we learned about the DNA and how does DNA replicate. DNA is basically the information that is in all your cells, and it actually is the blueprint of all life in your cells. But as information, and information can also be having errors, and the errors can cause a lot of consequences. So that's how mutation is. Here are the contents. We learn about the types of mutation, and here are the types: translocation, insertion, deletion, inversion, and then a bigger scale of it: structural change, chromosomal aberration, and lastly an even bigger scale, which is alteration of chromosome number. Definition of mutation: it is any changes in the structure or number of chromosomes in a cell of organism. It can take place in any cell of an organism, and because it is a blueprint, right? If you change the blueprint, then the product will be different. Let's say that you have a recipe, right, just like DNA, and then somebody erases some ingredients and rewrites it with something else. So based on the wrong recipe, you'll create the wrong type of food. And if this happens in the genes, okay, it actually causes the protein to be malformed. And if the protein is malformed, maybe the cell will be malformed, okay, and then tissue organ system might also be wrong, not like normal one. So if the mutation happens in gametes, then it can be passed down to the offsprings. In the scale of mutation, you have the smallest, which is gene or points, and you have the biggest, which is chromosomal. But the source, it can be either spontaneous or induced. In the cells, you can say that mutation can happen in the body cell or somatic cell, which is just the individual who is having the mutation, he will carry alone. But if you have mutation in the germline mutation, it means that the mutation can be passed down to the offspring. Spontaneous mutation. This one occurs naturally, just like the name says. It just happens. Nobody knows what is the cause of it, and it happens around 10 to 10 minus 4 to 10 minus 6 mutations of a gamete and gene. But for induced mutation, induced means that you have a cause, something that is something that you are exposed to would change your genes and mutating it. It is caused by mutagens such as radiation and viruses and also chemicals. So the thing is, this might be also done for good. Scientists, they induce mutation to treat organism with mutagens to see what kind of cell that you would create. Okay, mutagen, muta means change, gen means something that creates, so it is the cause of mutation. These are the substances or agents that can cause a much higher rate of mutation. So the process of mutation is called mutagenesis. So you have two types: physical and chemical. Physical mutagens, you have UV rays and X-rays. UV rays will induce dimer formation with the adjacent pyrimidine base on DNA change through covalent bonding. What does it mean? It means that instead of, for the DNA, right, the bases, they would attach to the other side, right, A, T, C, G. But what if the bases, they bind with the neighbor? Instead of A, T, C, G, it will be like A, C, T, G. So it's so the ladder will be bent, okay. And then you also have ionizing radiation. Ionizing radiation will be radiation from something that can ionize, meaning that it is high energy. It is so high energy that it can break the DNA strands and cause your DNA to be broken, alter purine and pyrimidine bases, and this will make something that's called point mutations or deletion. Okay, so the difference is physical mutagens will be something that is from energy, right, electromagnetic rays like UV and X-ray. Chemical will be something that is from a chemical reaction, not energy. Any chemical that can cause mutation are called chemical mutagens. Okay, mustard gases, nitric acid, ethylene sulfonate, EMS, or ethidium bromide. Here are some examples of severe mutations. As you can see, this is a two-headed snake, a frog with many legs, and a lamb with also many legs. So here are the videos of the Chernobyl babies. I cannot show here because it might be copyrighted. Okay, so you can see the link. The link is in the PowerPoint. You can just click. Here are the Chernobyl babies. They have been exposed to radiation fallout for a long time. Okay, it starts with the mothers first. The mothers are okay, but they got exposed, and then they were in the mother's womb during this radiative fallout. They have become mutated in the womb. Alright, so gene mutation. It is a permanent change in the DNA or nucleotide base sequence that makes up a gene. So if it is just one gene that changed, we call it a point mutation. So the point mutation can change in a lot of ways. Okay, let's say you have A. What if the A become T? So you have base substitution. Let's say that we have ATCG, right? A and then T and then C and then G. That's our sequence. What if we add another letter between A and T? We add G. So AGTCG. Okay. What does it mean? It means that now you have base insertion. What about you have ATCG in the beginning, but now you have ACG. The T is gone. So that's what's called base deletion. And lastly, you have base inversion, where the whole sequence are reversed. ATCG becomes GCTA. So gene mutation, it is acquired during a person's lifetime or passed down from parents. So parents have that mutation first, and then that mutation somehow happens in the sperm or egg cells, and then the offspring have that mutation. So what does it mean? That because that person has been born with those mutated cells, it means that the mutation will happen in all of his cells for their whole life. You were born with that mutation, and that mutation is in your zygote. Okay, so as a zygote, you have only one cell, and that is the only cell that you have, the mutated cell. And this single zygote will create a lot of cells that you use to make your own body, to make a head, to make a body, to make an organ. All of these cells, they happen to unluckily carry that mutation. So due to that, all of your cells are having that mutation. So mutation that occur only in an egg or sperm after fertilization, we call it the de novo mutations. De novo means new. So for the mother and father, their sperm or their egg doesn't have any mutations, but after they combine to fertilization, suddenly there is mutation happens. So that's how we explain it. De novo is when we say that the genetic disorder happens, but with no history from the father or the mother. Now for base substitution. So this is caused by chemicals or malfunction of DNA replication, an exchange of single nucleotide for another. For example, you have A, and then it becomes another letter, right? A to C, A to G, A to T. Okay, so the most common is transition. Transition is exchange of a purine for a purine in the A to G, a pyrimidine for a pyrimidine, C to T. Transversion, you have exchange of purine for a pyrimidine, or vice versa, mean that from C to A or T to G. So here you can see, if one letter changes, there might be a possibility that you might end up with another protein or amino acid that you do not want. Let's say that your original was CAC, right? His. But with one letter change, CAA, now you have another amino acid. So what happens? Okay, you're building a Christmas tree using Legos. You use green blocks. Suddenly you misread the instruction. You use red blocks. So now your building, your Christmas tree, has all green except for one block which is red. Okay, so you'll be a little bit wrong. So the thing is, not all of these mutations are bad. Sometimes it happens, but it doesn't have any effect at all. We call it silent or synonymous mutation. Why? Because if you look at this table, right? UCU, UCC, UCA, and UCG all code for the serine amino acid. It means that no matter what kind of change or mutation happens in the UC, U, or C, or A, or G, it still brings up the same amino acid. So that's why we call it a silent mutation, because it doesn't have any impact on the amino acid that is created. This one is a little bit different. We have non-synonymous mutation. In this case, due to a single letter being changed, now the amino acid has become different. You might have the missense. Missense meaning that the amino acid becomes a different from previously encoded. Okay, but what about the nonsense mutation? For nonsense mutation, the mutation causes it to encode into one of the three stop codons, UAA, UAG, or UGA. What will happen? If you have a stop codon, the ribosome will not create any more amino acids after that point. You know that for proteins, the ribosomes will make link by link by link by link of amino acid until you have a long protein. Let's say that the nonsense mutation happened after 5 links. So 1, 2, 3, 4, 5, and then your ribosome will say, oh, I have to stop here. And then what will happen is that you have a short amino acid, a short protein. So this is quite dangerous because depending on where the nonsense mutation happens, the protein that is created might be incomplete. Okay, so it's just like you just add a random dot or period after a random word in your paragraph. For example, once upon a time, Aladdin went to the cave. Let's put a period after the time. Once upon a time, Aladdin goes to the cave. So it's like once upon a time is the first sentence. Okay. Now the sentence becomes 2 short sentences. Okay. So one example of a disease is called sickle cell anemia. In this case, there is this gene that is supposed to make your red blood cell. Okay. Your red blood cell actually have this protein called hemoglobin. Okay, you need it, hemoglobin alpha and beta. But because of one simple mistake, 1 point mutation in the hemoglobin beta gene, what happens is that the hemoglobin will become an abnormal version of itself called the HBS. So it is caused by replacement of base A by T in the 17th nucleotide of the gene for base chain of hemoglobin changes the codon GAG into GUG. You know, because you have DNA, right? DNA, when you transcribe the DNA, the other part will follow suit. But you have ATCG, right? Okay, so if you change the A into T, so the partner or the RNA will be from A to U instead. So now you will have a very different protein that what is supposed to be. So normally you will have CTC GAG, so it is for glutamic acid. But because of a single error from CTC to become CAC, now you have in the ribosome will match with GUG, you cause another protein to be created, valine. Okay. So insertion and deletion. Insertion and deletion is not like the substitution, right? This time you don't change, but you add or remove one base. So this causes an alteration of a reading frame and producing an entirely new sequence of amino acid. So the thing is, you have the letters going in like 3 by 3, right? A sequence of 3, 3, 3, 3, right? So if you have 4 of these letters, 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, okay, 10, 11, 12. If you add 1 extra, now you have what? 1, 2, 3, 4, 5, 6, and then you add 1, okay, and then 6, 7, 8, 9, 10, 11, and then 12. So now you have 1 extra. So the reading frame has already shifted. That's what you call a frame shift mutation. Okay. Let me explain through here. You have AUG, CGA, UUA, UAC, GGG. Okay. What if you add something else here? You add a U between this U and A. Okay. Add here. So now you get UUA. And then this C goes where? This C goes to the next word, which is CGG. And then this G, okay, where does it go? It's unpaired. Okay, it's just alone here. So from before this insertion, it is normal, but after the insertion, it becomes different. Okay, the amino acid that is created will be different. Okay, what if we delete one of the letters? We delete UAC. So when you delete UAC, then this will be empty. Who will go into here? So it will be UAG, and then GGA, AA. So this is dangerous because you can see UAG is a stop codon. Stop codons, when the ribosomes read it, it will stop doing or making the amino acid. You have AUG, CGA, UUA, and then suddenly UAG stop. Okay, and then the ribosome will disengage. So you will have a short protein, a short amino acid, which will make a smaller protein instead. So for example, this is a disease called cystic fibrosis. So this happens around 1 out of 2,500 white births, white people. So this is a disease that makes your mucus, whenever you sneeze, you have this snot coming out. Well, how does it feel? It feels like liquid, right? Imagine if that liquid, they are very sticky like glue. Okay, they're so sticky like glue, right? It will kind of like block your nasal passage, block your breathing tubes, bronchi and bronchiole. Imagine if your mucus is thick as glue. How can you breathe? Okay, you cannot breathe at all. So the thing is, due to a mutation in terms of deletion, it actually causes this salt channel or natrium channel to be broken. So there was this gene that is supposed to code for protein channel that allows salt to flow across cell membrane. Okay, when the salt flows through across cell membrane, then osmosis will change and it will suck water to make the mucus much more viscous, much more flowy. Whenever you sneeze, it's like watery, right? It's not like gluey, it's watery. So imagine if that gene doesn't work, so the cell cannot create the salt channel. So salt cannot get out of the cell, water also cannot get out of the cell. So the mucus will be thicker and stickier around cells. And due to them becoming quite sticky, they're unable to go anywhere. They will be stuck inside of the lungs. Okay, because they are like glue, they are supposed to trap all of this bacteria so that the bacteria will not affect our lungs, right? But because they are too thick and too sticky, it will trap all of the bacteria. The bacteria cannot go anywhere because the mucus are stuck there. So the bacteria lives there. So it causes bacterial infections. So due to that, it will destroy the lung function. So yes, it is really hard to breathe when your lungs is filled with glue. Okay, so without treatment, children will die before age of 5. With treatment, maybe they can live past the age of late 20s. So here you can see this is chromosome 7. There is a gene called the CFTR gene. This is the gene that codes for the salt channel. So within this sequence, you have ATC, ATC, TTT, GGT, right? And the part that is deleted will be CTT. Okay, what happens that when this is deleted, there is no phenylalanine 508. So one amino acid is lost. It causes the salt channel to not work. As you can see in this picture, these are how the mothers have to deal with every day. They have to tilt the babies, tilt the children, so that the mucus will slowly flow to the throat so that they can cough it out, because the mucus is so sticky. It's very hard to breathe. And in other times, the mothers have to put them on their lay them on the front like this, and then have to pat them so that the mucus will come out. So here it is. Original, what will happen here? This is like you are adding something, right? This is like you're adding something. Okay, this is also addition. This is deletion. And this one is a lot of addition coming here. Okay. So now we move to something that is called a frame shift mutation. So in frame shift mutation, you have base insertion or deletion that actually shift the frame of reading. So I mean that the reading frame, your ribosome reads the base as trees. three letters every time. If you add 1, then you have 2, which will be frame shifted to the other word. So meaning that you will disrupt the reading frame or grouping of the codons. It will cause codons to be read differently. Due to that, the amino acids after the frame shift will become a different type of codon that will code for different type of amino acid. And if you somehow get a premature stop codon, UAA, UAG or UGA, what happens is that this will cause the ribosome to stop. Okay, and protein that is created might be abnormally short, long, or contain wrong amino acids, or not functional. Certain sequence of nucleotides encodes a different sequence of amino acids. You can see here, GLU, PRO, GLN, LEU. Okay. What if this frame shift mutation happens? Now we have AGC, serine, CGC, ARC, AAC, ASN, UUC, valine. 4 totally different amino acids compared to the original. Now we move to the base inversion. The base inversion involves the reversal of portion of a nucleotide sequence. Connection between genes break and the sequence of these genes are reversed. In the new sequence, it might not be viable to produce a gene product. So you have ABCDF GHI. If it is inverted, so it is FED GHI. So original sequence, and then the inversion, you have TGC A becomes ACGT. So now we move to a bigger version of it, chromosomal mutation. There are two kinds. You have chromosomal aberration structure or number alteration. This occurs during the condensation and the separation of chromosomes during mitosis or meiosis. Maybe it can also occur during the replication of DNA in interphase. You still remember the cell cycle, G1, S, G2 and M. Okay, maybe during the G2 replication in interphase, there might be a little bit of error or something. Crossing over in prophase 1 also can make it happen. So these are the words that you might have been familiar with: deletion, duplication and inversion. But there's actually one more word that is new, which is translocation. So this one is a little bit different, but let's go to it one by one. Deletion is just like the point mutation. It just involves segments of DNA being deleted. You have micro deletion, terminal deletion, and intercalary deletion. So if you can see in this chromosome, you have segments A to G, and then if C and D disappears, now you have A, B, E, F, G. So not enough. Terminal deletion is a deletion that occurs toward the end of the chromosome. Intercalary, it is in the middle, interior of the chromosome. Macro deletion is the small amount of deletion which is less than megabases. So gene is indirectly removed from the chromosome, which causes loss of genetic material, and abnormalities and serious genetic disease. Small deletion, not really fatal, but large deletions mean that you have lost a lot of genetic information. Usually it is fatal. For example, this is a mutation called the Hybridization syndrome. It's French. Deletion on the part of the short arm of chromosome 5. The name itself in French means the cry of the cat. It's because the babies who have this Hybridization syndrome, they will cry like a cat and not like a human. Also, they will grow up with severe mental retardation and shortened lifespan. Inversion. Inversion is the rearrangement of chromosome segments. Structure of the chromosome is reversed or inverted. Segment is cut at 2 points, inverted and 180, and inserted back into the same chromosome. So there are two types: paracentric, that does not involve centromere, and pericentric, which involves the centromere. Now for duplication. Duplication will be doubling or multiple copies of the chromosomal regions. It involves the insertion of extra copy of a region of chromosomes into the neighboring position. For example, the Charcot-Marie tooth. You can see this is a segment. Segment has been doubled, duplicated. So now you have two segments. Translocation, it is when a chromosome segment shifts to another region within the same chromosome or another chromosome. If it happens in the same chromosome, it is intrachromosomal translocation. But if it happens in another chromosome, it's called interchromosomal translocation. For example, the family Down syndrome or translocation Down syndrome. As you can see, this is the red and blue chromosomes. They both break and they exchange places. Now you have translocation chromosomes. So here, chromosome 4 and chromosome 20, right? Chromosome 4 is really long, chromosome 20 is really short, and they exchange their segments. So now you have chromosome 4 with a part of chromosome 20 and chromosome 20 with a large part of chromosome 4. Reciprocal translocation is when the two homologous chromosomes break and exchange fragments. It means that each one of them lost and gains a new segment. Kind of balance, right? Non-reciprocal is not balanced because one will lose its segment and the other gain segment. So it is not balanced. It's one-way transfer of a chromosomal segment. Chromosomal number alteration, this one is about the number of chromosomes. Chromosomal number alteration is about the number of chromosomes. It is due to errors in meiosis or mitosis. There are two types, aneuploidy and polyploidy. Aneuploidy is one or more chromosomes from a normal set of chromosomes that are missing. So they are present in an unusual number of copies. So either they are extra 1, extra 2, -1 or -2. So the organism, if they lose or gain the chromosomes, they are not fulfilling the 2n. So remember that every organism must have an even number of chromosomes, which is 2n. If they have extra or if they have less, then it will not be the same. Then it will result in autosomal or sex chromosome abnormalities. For monosomy, it is a loss of one chromosome from a set. You know, every chromosome have their partner, right? Chromosome 1, 1. Then chromosome 2, 2. Because you know, one for the father, one for the mother. If one of them is lost, now you have a single chromosome. Call it monosomy. So nullisomy is the loss of two chromosomes from the diploid chromosome set. So now you have both of the chromosomes, both of the partners gone. Okay. Trisomy, you gain one extra chromosome. Tetrasomy, you gain two extra chromosomes. Why is it tetra? Because tetra means 4. So now you have 4 chromosomes of the same kind. Double monosomy will be loss of one chromosome each in 2 pairs of homologous chromosomes. In that you have chromosome 1 losing 1, chromosome 5 losing 1 also. So you have monosomy happening twice. Double monosomy. Double trisomy is like the trisomy, but it happens in 2 chromosomes. So now you have 2n plus 1 plus 1. So now some chromosomes have 3. Another chromosome have also 3. Okay. Double tetrasomy will be 2 extra pairs of homologous chromosomes in the diploid chromosome set. So you have chromosome 1, it originally have 2, now you have 4 chromosome 1. And then maybe chromosome 16, start with 2, now you have 4. Okay. So you have 2 extra pairs of chromosomes in 2 sets. This is what you can see here. Diploid is the original. This is nullisomic because here both of them are gone. Here is monosomic, only one is gone. Here is double monosomic because this one is gone. This one, which is 3, also gone. So it is gone from 2 pairs of chromosomes. Next is the trisomic. 2n plus 1, which is this chromosome of 4, has 1 extra. The trisomic will have, okay, this pair number 4 have 2 extras. Double trisomic means that there are 2 pairs that has 2 extras each. So 3 is supposed to be 2, now you have 4. 4 also, you are supposed to be having 2, but now you have 4. Okay. Aneuploidy. Aneuploidy, it is usually happen due to chromosomes fail to separate during anaphase 1 or anaphase 2. So this non-separation we call the Nondisjunction. So some cells have too many or too few chromosomes. So during anaphase, you know that homologous chromosomes will move to the different part of the cell. One goes to another pole, one goes to this pole. Okay. What if both of them move to the same pole? Then you have extra chromosomes. During anaphase 2, gametes which are formed will contain either none or both homologous chromosomes. Okay, here you can see in meiosis 1, right? Here is normal, but here these two doesn't want to separate. Okay, so now the only one that is ending up in this cell will be 1, 2. Okay, just 2 chromosomes. But here you have 3, right? 1, 2, 3. 3 chromosomes each. Okay, from here, in meiosis 2, now you have extra n plus 1. N plus 1 extra. In here, because it has lacking 1 chromosome, the 1 chromosome has gone to the other side, so now you have 1. So now you have n minus 1. Okay, but if this non-disjunction happens in the second meiosis, then there will still be a normal cells. So you can see in here doesn't happen yet, and then they divide. And here, what will happen? Here will be normal, but here, because these two doesn't want to separate, one have extra, one have less. For example, trisomy or Down syndrome, it is also called Mongolism, and because back then people used to say that these Down syndrome people, they look like Mongols, but now we do not use it anymore. But still, it remains as an insult for Brits and for Aussies. Sometimes they see that their friends are acting really silly, and they would say, "Yo, why are you acting like a Mong?" Okay, so do not say that because it is really insulting. Okay. An individual with Down syndrome has 3 copies of chromosome 21. Okay. This happens in Down syndrome. It occurs in 1 out of 800 live births, and the probability to get Down syndrome increases as the mother gets older. So for the mother who is young, they'll be less likely to have this Down syndrome. Here, as you can see, it is non-disjunction in meiosis 1. You have extra pair. This one has lack of pair. Okay, so due to this extra pair, if this happens in chromosome 21, then you'll have your Down syndrome. So this is the karyotype of Down syndrome. Okay. What is the odd one? Here, chromosome number 21 has 3 instead of 2. Okay, 1 extra. So for Down syndrome babies, you can see that they have flattened nose and face, upward standing eyes, single palmar crease. Okay, look at your palm. How many creases are there? Oh, the camera doesn't catch. Okay, here. 1, 2, 3. How many lines? 3 lines at least. For them, they only have 1, and 5th finger is short and curved inward. So your 5th finger should not be that short. And then this is another characteristic of the Down syndrome people. Widely separated first and second toes and increased skin creases. So Down syndrome is associated with some impairment in cognitive ability, physical growth, and facial appearance. It can be identified during pregnancy or at birth. So you have symptoms such as poor muscle tone, so we call it muscle hypotonia. Protruding tongue due to small oral cavity. Due to small oral cavity, means that they have a small mouth. Okay. Enlarged tongue near tonsil and short neck. But it doesn't mean that they are non-functional. They are actually able to become adults. A functional adult, they can do work, they can do jobs. It's just that their mental age is a little bit a few years behind from their actual age. Imagine like somebody who is 21 years old, they act like they are 16. Okay, thinking about girls, thinking about playing games. Okay, somebody who is like 11 years old but act like a kindergartner. So that's how Down syndrome works. Still, when they become adults, they are just normal. Okay, they can go to work, they can count, they can understand what is in TV. Okay, maybe they speak a little bit slowly, but you know, they are still functional. Alteration of sex chromosomes. They cause developmental errors. It is specified either male or female. This is a result of non-disjunction during spermatogenesis or oogenesis. For example, Turner syndrome. Turner syndrome, the individual is born with 1 sex chromosome and X. Individuals with Turner syndrome are 45XO. Sexual characteristics are present but generally underdeveloped. So it means that they are infertile. They look like women. Outwardly, they are like girls. They can, you know, they are able to be born. They are able to live. It's just that they don't have any ovulation, and it means that they cannot have any children. Other than that, they also have a short stature, low hairline, shear-shaped thorax, short-term metacarpal 4, part of your hand bone, elbow deformity, repentic ovaries, means that this ovary just appears, but it doesn't do anything. No menstruation, poor breast development. As you can see, in terms of their chromosomes, they are missing one. Okay, they are supposed to be an X here, but they don't have it. Okay. Now for Klinefelter syndrome. Klinefelter syndrome people, they are males who inherit an extra X chromosome, so they have XXY. Okay, so because of this extra X, they become much more feminine, high-pitched voice, gynecomastia, which is the breast development in males, little facial or body hair, sterile, maybe subfertile, small testes or prostate glands, and low testosterone. But still, they are male. So here is their karyotype. They have one extra X chromosome. So another one is Jacob syndrome, which is called XYY syndrome. So in this case, a man inherits an extra Y chromosome. So this is also called the super male phenotype. The Y chromosome comes with the extra testosterone, extra maleness, so it causes severe acne, fertile. Okay, they can still have children, right? Above 6 feet tall, very tall, and produce high levels of testosterone. So here is a confession of an XYY male. So here is Ed. He is a pathologist. He has an XYY chromosome. He is tall, lean, physically powerful. Okay. Even at old age, he has to take medicine for the pimples, acne. But being an XYY male, extra testosterone, he has this temper, and he is controlling his temper. So he has controlled himself to avoid any situation that would set his anger off. So he has never physically hurt anyone in the anger. So here is a man who has been born with extra testosterone but still is controlling it. Okay, so he has stronger muscles, due to the effect of testosterone. It builds up your muscle. Okay, so he's much more strong compared to coordinated. Okay, so he's much more focused on strength in different sport like swimming or gymming. So the thing is, in terms of his thinking, his cognitive, he is a little bit different and always has been, but doesn't bug him. So there's a big prejudice that XYY people are criminals. They have tendency to do violent things. They tend to be serial killers, but actually that's actually a myth, because as you can see, an XYY male, despite born with more testosterone than a regular male, they can also behave. They can control their emotions, keep them in check. Other than that, you also have the super female trisomy X chromosome, trisomy X syndrome. You have three X chromosomes, so they are taller than average, long legs, normal sexual characteristic, but lower range of normal intelligence. So here is Gemma Ward. She is a supermodel. Many people have thought that she is a XXX chromosome person, okay, having a super female phenotype. Okay, she has abnormal ovaries and breastbone, taller than average, and an abnormally curved pinky finger. So a lot of people would say that she is a XXX chromosome person because she doesn't look she supposedly look taller than normal for her own age. Polyploidy. It is a state where the number of set of chromosomes exceed the diploid number by a multiple of n. This happens due to the failure of spindle fiber mitosis or meiosis to segregate chromosomes into separate groups. So this is called Nondisjunction. So you have euploid. Total number of chromosomes is a multiple of haploid number, which is n. For example, you have diploid. Okay, diploid is normal. So double the number of single set of chromosomes. Triploid, now you have 3 sets of chromosomes. Tetraploid, you have 4 sets of chromosomes. Even more, you have pentaploid 5, hexaploid 6, octaploid 8, decaploid which is 10. How does fertilization happen? So you have diploid, fertilized with normal N gametes. You have triploid. A diploid just fertilized with diploid. Now you have tetraploid. Usually you don't find this in animals or humans, you find it in plants. So the more chromosomes, the more ploidy, they will actually become much more bigger and taller in terms of the growth. So there are two main types, autopolyploid and allopolyploid. Autopolyploid will be the additional sets of chromosomes derived from the same species. They arise from a spontaneous, naturally occurring induced genome doubling of single species. You have from A to AA, or maybe 4 A's. For allopolyploid, it is a little bit different because you now have chromosomes which are from two origins, different species. You add them both together. Now you have hybridization and doubling of genomes from different species. So now you have A and B. Okay, you double them, you have AA, BBB. Okay, and then you have C and D also, CCDD. Now we have CCCDD, meaning that this plant actually has genes and chromosomes from other plants. So now they are super plants. So somehow it works in the world of plants, but not in the world of humans or animals. So that's all for this lecture. So I hope you all have known what mutation is, as simple as it is, and what causes it, and what are the types of it. So I hope you all have a good day, and thank you. The end.